My pleasure to introduce our speaker for the 2025 start of our CMM seminar series. Our first speaker is Professor Sid Goyal from the Department of Biophysics here at the University of Toronto. And he will be talking about competition in biology across scales. So real multi-scale, complex systems kind of talk.
Absolutely.
Should be great.
Thank you. Thank you. It's fun to, it's interesting. For a while I had no interface with Field Institute, and this year, how you count the year, but I've had a chance to talk to a lot of you guys, which has been exciting. But it was also a bit of a challenge because I didn't want to give the same talk I gave last time, which was around tumors. So I wanted to tell a little bit more besides tumor, what my group does, and how I sort of think about how it threads together in some ways. So again, we are a small group, even Zoom, so just interrupt whenever. And I'm looking for an hour. Okay, so yeah, just ask whenever, whatever comes to your mind. And yeah, we are in physics, just right across. So, that's also a good reason to have some community, local community. All right, so I just wanted to say, I mean, this picture is kind of a weird one, but I wanted to highlight competition in some ways and stochasticity. So those are the two ideas that are going to interplay a lot. So there's a lot of chance play in, how things play out, but there's also sort of, yes, sir.
So this collaboration
also
is complementing competition.
Sure, yeah. So I'm very good, very good. So the competition could be competitive or it could be collaborative. So I'm using a little bit sort of right. Well, the styles are merging very fast, so, so I don't know. Yeah, exactly. So I don't know how that's going to play out, but good. So, so I also wanted to say I do a bit of experiments, which I'll also talk about, but I also do, 60–70% of my work is more theory. So I just wanted to put a sort of a little bit of a conceptual spin on it. The idea, of course, in biology is the objects are pretty complex, right? And, this is a famous picture of Picasso, the bull on the left, and there's a sketch of it. Is there a pointer I could use? Let's take maybe. No. Oh, okay. No, never mind. Right. So I could use it. If you don't mind it, I'll just use it. Right. So I mean, this is the bare bones of that bull, right? So I don't know which direction Picasso was going, but it's very clear to us when we want to write down theories, we want to go from here to here and find some description of this kind, which is just enough to whatever we want to explain. It doesn't capture everything, but it captures hopefully the important bits. And I'll just flash a figure from an old piece of work. So this was some data we were looking at, a long time ago now, on how blood regenerates in monkeys, right? And this is the kind of data you get, which is pretty, I mean, at the face of it, pretty horrible. I don't know what to make of it. What am I going to compute from that, right? But it took us some time to realize that we can replot this data as distributions, and this is how it looks. So what this data is basically slicing around this axis and making distribution at different time points. So I can go at time 5 and find sort of a density function, if you will, of what size the clones are. And I get a cumulative distribution which looks like that. And I can do that at different times. And I get this beautiful curve, which is invariant in time, while the individual clones are going crazy, as you can tell, right? Now, this is something we can work with, right? This is where if you gave me this data, I would be ready to write down some theory for you, right? Not here. But I think there's a lot of in biology where we get this. And I think the first part of theorist happens to be, actually trying to figure out how to plot the damn data, right? You can plot it right, then you can start doing some theory, which could be interesting and predictive and, all the good things we like about theories. So I think there is this sort of important point in biology which remains, I think, it's an old thing, but what are the right coordinates that you want to look at the data? I think that is a fundamental problem in how to attack biological problems, and I just wanted to put that right in the center. And many of the things we do revolve around that. Competition. Okay, so I just wanted to say that competition we can think of in many ways, But competition and evolution is something which kind of goes hand in hand in my view, and this is how I want to think about it for this talk. So I just wanted to lay it out. You can think of competition as, having a population of cells, individuals, whatever your scale you're interested in, and they can have sort of progeny in time, some more, some less. So this is the chance element of it, right? Some might not have any progenies. Some might have more than one. Some may just have one, and so on and so forth, right? And once in a while, one of these individuals would change, right? And this change, usually we think of it as a genetic change, as a mutation. But this could be an epigenetic change. This could be a change in how cells decide to express its proteins. So, any kind of change, if you will, which, if heritable, can then lead to sort of this cell, for example, having more than its share of the final population. In this case, for example, it has a slight benefit in terms of, in this situation, right? This could be a tumor cell. This could be a microbe under antibiotics, whatever context is interesting, right? So I just wanted to say, this is how I want to think about competition. And then you can go in the end and look at, for example, if this was mutations, you can sequence a bunch of them and figure out, you can go back and infer many of the things when these mutations might have happened, because they might have all happened in the background of this particular lineage, for example. And you can go and place them, when would these mutations happen? For example, this light blue one seemed to be in all of them. So it probably happened early enough. This one is in the background, which is not this. So this might have happened anywhere. I don't know where it happened somewhere, right? So you can do a lot of inference like these. So I wanted to talk about evolution, genetics, phenotypic diversity, and this is the spirit in which I'm thinking of competition and cooperation or whatever. Again, people can do, this is some data from, I think, sorry, I forgot the reference, but this is some actual data people plotted from some experiment, right? It's sort of smoothened out and things like that, but you can get very rich data sets, and again, microbes are the place where people do a lot of it, protein evolution, bacteria, viruses, I should add COVID to it, antibiotic resistance, and so on and so forth, right? So what do we want to do? So in my group, we've thought about this sort of competition, if you will, on different scales. So I'll probably try and give you 3 examples, depending on how we do on time. The first one is actually at the barest genome scale, which is what we expect things to be. But here, what is interesting is this is a yeast cell. So all eukaryotic cells not only have nuclear DNA, but also have DNA in its mitochondria. So now within a cell, you can actually have a diversity in your mitochondria, right? Because this mitochondrial DNA can mutate. You have multiple copies. You have hundreds of copies, sometimes thousands, sometimes 10,000, right? So our cells can have many, many copies of mitochondrial DNA. So within a cell, you can actually have this whole competition going on within a cell, right? So we wanted to understand, in yeast, this is a known phenomena. We wanted to understand what happens to these genomes as they mutate, and can we now assign fitness to a genome, not to a cell, but to a whole genome. And they make these weird repeated structures, which we sort of sequence in single cell, single molecule sequencing, and you can kind of think of this structure giving it fitness, right? I'm going to talk about genome structures and how their replicative potential depends on that. That's the smallest scale, if you will. The second obvious one is microbes, bacteria. Here we thought not just of bacteria, but again, bacteria and viruses which kill bacteria are sort of everywhere, right? They interact all the time. And if I think I have a slide about it. This is precisely how the food web starts, if you will, right? Phages killing bacteria in deep ocean is where all the food is getting assimilated, and a lot of this sort of cycling happens there. So there's a constant competition between phage and bacteria. And one scheme bacteria has to save itself from this is exactly like what we have. When we get vaccinated, we remember our past infections. So bacteria can actually remember past phage infections. They actually integrate. They have a small memory of what the past infections it has encountered, and remembers these and uses them to sort of mitigate the next attack, right? So it's a bit of a learning problem, if you will, right? So they kind of learn and remember what the external world looks like, and that helps them sort of mitigate future. So you can do a bit of problems there. The third and the final one is basically, so these are both genetic, some level, right? Here, it's not genetic in the sense it's not waiting for a mutation, So it's already sort of going away from that. It's learning, it's trying to remember stuff, but still it integrates stuff in its genome. This stem cells and tissues is bona fide non-genetic, right? So if you start with a population of stem cells in, let's say, in our bone marrow, all of them are genetically identical, but many of them have different fates along which they go. Some make more red blood cells, some make more white blood cells. So there's a bit of chance and competition going on between those cells on what happens. And some of them might actually become tumor cells and really outcompete the others, right? And some of that is genetic, but there's a good component of that is also epigenetic. So I'm going to think about that as a non-genetic diversity and how that plays out in the problem. So those are the three problems I'm hoping to address. We'll see how it goes. I'll start with the stem cells, because I think that's sort of the phenotypic part is what we usually don't get to hear much about. So I thought it'll be interesting. It's a famous picture by Waddington. So I'll say more about it precisely in like 10 seconds, right? So think of stem cells, right? So this is, let's say, your embryonic stem cell. This will kind of make, progenitor for blood cells and skin cells. These will then make, your differentiated cells, as you know. But all of this lives on some weird landscape, right? And this was famously drawn by a guy named Waddington, right? Who was basically, even before we had a good microscopic picture of what was going on, he's saying, look, I mean, this one cell is having two different fates. It really is going down some path, bifurcations, whatever you want to think about. And underlying this is all those sort of little genes and molecules, whatnot, controlling this thing and pushing and pulling these things. But there's some landscape on which things flow. I mean, I have to say this is a bit of a metaphor, but it kind of has been helpful. This concept has been quite useful in thinking about formulating problems in this way. I don't think we still have there's only very recent couple of papers which have really taken this at complete face value and tried to write down simple bifurcation models or catastrophe theory type ideas when you see these different cell states, what kind of landscapes there might be and whatnot. But still, it has been a very interesting way of thinking about it. But there's more to it. You can actually take a skin cell and reprogram it back to an embryonic stem cell. It's usually called a pluripotent stem cell or induced pluripotent stem cell. And you can actually make the whole organism from it. So you can actually take a skin cell of a mouse, make an egg or a sperm out of it, and then fertilize it and make a whole new mouse out of it. Right? So you can actually do the whole thing. So this reprogramming is very, very powerful, right? And I think there was some sort of ethical issues about it, which people tried and this and that. So, well, we can go into all of that, but yeah, lots of things. So basically, yeah, pluripotent cells, which people are now, you can store your cells when you're born, right, from the placenta, and they can use that to kind of do bone marrow transfers, for example, right? You can kind of, so we have done some work along this. This was the old work, which started a long time ago, which a couple of people at UCLA when I was a postdoc, and we were thinking of this axis, blood, right? In Toronto, and this was sort of the figure I was flashing earlier. So very similar ideas kind of helped us think about project we did here with folks at U of T, now also a few years ago, on thinking of reprogramming, right? And I'll formulate the problem a little bit better. And the tumor stuff is what you guys have, some of you have heard, which also had sort of a similar idea of, what is happening in the middle? Because this is great, this is all developmental, but there's all these pathological directions and cells that can come about, and how can we think about those? So I'm going to start with cellular reprogramming. That's the project which we did here with Toronto. Sophie was a graduate student, now happily graduated and doing other things, but she led most of the theory work, and the experiments were done with a colleague in bioengineering. So the idea was we wanted to think about skin cells. But if you really think about it, a skin cell and a pluripotent cell are actually quite far apart on a gene expression space, right? So if I look at, I take a skin cell and I take embryonic stem cells, the number of genes that are different between them are hundreds, thousands, right? Many, many different genes are getting differentially expressed. Now you tell me I'm going to take a skin cell and do something to it, and it's going to become this pluripotent stem cells. That seems a little bit too magical. And it was. It was, you know, this was done in what, 2000 and, I'm going to get it right, 6, I think, 6 or 9, and the person got a Nobel Prize 3 years after, right? Because it just completely revolutionized how we think about plasticity in cellular behavior and development and whatnot. But you still have to go from this end to this end, right? This is happening, so we wanted to understand that. And the idea in the field has been for the longest time is that, look, all these skin cells are genetically identical. They're all from the same mouse, right? So all of them has some chance of making it to this, right? The chance might be low because, you know, they kind of have to traverse some weird path in high dimensional space, but it happens, and they're all equally likely. But it's not obvious there's only one path to this, right? There might be multiple paths going through this process. Because again, if it's a search problem, well, first you should be worried that I'm saying it's a search problem in 100 dimensions. I mean, it's not going to happen, right? 3 dimensions is already pretty hard to do a search from one end of the room to the other. So, you know, 100 dimensions seems weird. So there's something more definitely is going on. But people knew that there are some intermediate states they could characterize, and many of the cells seems to be going through those. So the question we wanted to ask with these experiments was, you know, is reprogramming pathway, which is going back, kind of similar to development in the sense that development, how does development work? You can say, look, you take an embryonic cell and makes all these different cell types. How does it know that? And we know that it takes almost the same path. Every organism, every baby that has a heart almost has a canonical path of how cells start from embryo, and really there's a dedicated path where these cells are handheld with external morphogens. Everything has to be just right for them to get to this particular state. This is in a dish, right? There's no morphogens. There's nothing from the outside when this thing happens. That's why the probability of this is pretty small. You know, you get a very few cells, but when you get a few, it's a jackpot, right? But the question we wanted to ask is, well, is there some signaling going on which we are not seeing? If you reprogram a single cell versus a population of cells together, you see some new effects, right? Is there some collectiveness to this movement? So we can make some mean field models. So what the next slide is. So yeah, you can make some mean field models of going from skin cells to iPS cells if you kind of keep track of how many cells you're getting of different types. So you can imagine you can define states of cells and look at them in time and how many you're getting. But what we were interested in is the heterogeneity across the cells, right? So we wanted to have a population of skin cells. They're becoming these pluripotent cells. And what I want to understand is, how does the contribution from individual cells differ? Okay? Some of it is going to be just stochastic because the cell just died, you know, on way of becoming something. But how does this process unfold at the single cell level? So what Nika did was actually put genetic barcodes on each of these cells. So now you can track lineages of these individual cells as they are becoming these. Because you can go at any time point, take a bunch of cells, sequence, and that tells you how many cells came from this yellow cell, how many cells came from the red cell, and so on and so forth. Right? So now you have a single cell view, clonal view of how each cell is populating this new population, right? So now you have a very high dimensional view of this thing, right? And the experiments were done from day 0 to day, so it's a 30-day protocol, and these were the times things were sequenced. So what do we look at again? Well, we can make those cumulative distributions I just told you about, right? I don't still know how to think about individual temporal trajectories, but we can make those distributions, right? Sorry, I flipped the cumulative from going up to going down. But so this is day 0, day 8, day 4, you know, and so on and so forth. You can plot these cumulative distributions, which are these solid lines, right? Sorry. So I guess, but so the question is, well, what's the full model? What do I expect, right? If things are completely stochastic, right? What I mean by that is you just flip a coin every time you have to change a state, and you get as many cells, you know, like the neutral process, neutral evolution as you would imagine. The distribution should look like this dashed lines. That's what you would expect if there was no one cell better than the other, right? By chance, some cells will divide more, some will less, but that's what it is. So day 0, you actually look at these cells. So you barcode everything, and allow them to proliferate a little bit before you have given these. When you're turning skin cells to iPS cells, you have to give them something, right? You kind of excite them with 3 or 4 transcription factors, which are the proteins which allow change the gene expression of these things. And you get a distribution which really looks like a neutral. So these cells are dividing without really having much difference in their ability to divide. But these distributions become quite different from the null distribution as the time goes on. So the stemness is coming from, in the sense, these cells have a, so the question is a little bit deeper. You're saying what defines stemness, right? What is the difference between a stem cell and a differentiated cell? So in some ways, the answer is boring. From a quantitative standpoint, there are specific genes that are expressed and specific genes that are suppressed, right? And when you have those things expressed, the cells are pluripotent. They can actually go around in many different directions. As you can imagine, basically what happens in a stem cell is it has many more regions that are open to be expressed, but it controls them pretty tightly. Absolutely. So in the end, there's some genes which are expressed more, some are suppressed, and there is some way that plays out that the properties the cells have are such that they can now, if you give them the right signal, it'll become anything. So if I take my skin cell right now, I can give it whatever it wants, it's not going to become a neuron, right? But I can take a skin cell, make it a pluripotent cell. Now I can give this cell some signals and it'll make a neuron. It'll make a heart cell. It'll make a lung cell, whatever you like. Right? Good. So the first question is you start seeing this. So this immediately says, well, they're not neutrally dividing. You know, there's somehow some cells are better than the others, and you know, they can be better by a whole lot. So wrong laptop. So what we said was, can we write down the first simplest model? So the first simplest model is, well, there's some fraction of them that does better than the others, right? And now you can have a small elite population which reprograms much faster than the other. It has some fitness in terms of, you know, how fast it reprograms. And with this one parameter, so this is sort of a 2-parameter model, right? The 2 parameters are, what is the fraction of elite cells that they are in the population, and how fast do they grow relative to the rest of the population. So with this 2-parameter model, you can actually get the solid lines pretty accurately now, right? In time, right? So you're like, okay, fine, you should not be too impressed. It was almost a curved line, and I took 2 parameters to fit it, so it should not be too surprising, right? The interpretation is a little bit interesting, but it makes a strong prediction. The strong prediction is, if I'm telling you some cells are better than the others, right? Now, if I did a split experiment, the same cells should become big in both of them, right? So there should be high correlation between different cultures, right? Because it's not stochastic anymore. So this is what Nika decided to do. She basically split them and find these 2 things. And now you can look at how things are correlated in time in these 2 cultures, right? So x-axis is the size of the clone in one culture, the y-axis is the size of the clone in the other culture, right? So this clone, which is bigger in both of them, clearly has to come from some fit clones, right? So now these correlations you can compute, right? And the theory I wrote down earlier then predicts these correlations without any fits. So these were 2 different systems, and this the bars are the calculation from the data, and the gray dots are predictions, right? From the model from the previous experiment. So from the previous experiment, I compute 2 numbers, and in this experiment, I just test those 2 numbers, how well they can predict something for a new experiment, right? So now the model is predicting something which you didn't put in by hand, right? The first slide was just fitting. Now we are saying that this view that you have some small fraction which is better than the other seems to be holding up in a different type of experiment. And you can even do more. This can also predict sort of how fast the fraction of unique clones die. As I said, some clones die out, right? And now if some clones are better than the others, the death is going to be much more, because those clones are out competing the others, right, compared to the neutral ones. So this sort of two population model also predicts these trajectories very, very well. So now these are not fits. These are just predictions and a completely different experiment from what we got. So I just wanted to highlight an example that you can make simple models and extract a few parameters, but then the challenge is to really go further and test some of those and see if they hold up. So I think this has been interesting to figure out the way this reprogramming works, and you have this elite population which is genetically identical, right? Again, there's no genetics going on here, right? There's something special about some cells, and you're hitting with a hammer. You turn on some 4 transcription factors at some high level, and you let this thing go. And somehow some cells manage to reprogram thousands of times better than the others, right? So these are log scales, right? So these are log scales. Nothing. They are genetic. They extracted from the same pool of mouse. You just extract them, barcode them, and you let them go, right? I mean, clearly there must have been some early gene expression differences and things like that. But what I'm not showing you is that you can actually, this is because it's a temporal analysis, you can go backward in time and ask which time is predictive of what you see in the end. And it turns out that it's not at day 0. It's not that the cells are already different at the start. Somewhere around day 8, day 14, some cells figure out the right direction to go, it looks like, and then they zoom past. So that gives us a new hypothesis, right? So we think that there are actually highways which can go from this end to this end. The cells have to find those highways early enough. So some cells find those highways early enough, they zoom past. The others are doing this search problem, and some might find this later, and so on and so forth. So that completely changes the view in which reprogramming works. And now we have been spending time trying to think about how to find these highways from this new kind of data. This data is not enough because highways are in gene expression space, right? So we're trying to develop ways of thinking about that. How am I doing on time? Okay, another half hour, right?
They need to hit the point. Then you have to get 3 genes.
Something, yeah, exactly. There is a path. There's a path. Exactly. There's a path, right?
Hypothesis, couldn't it be
Yeah.
genetic markings? So they're the same DNA, but
Correct.
they're the same genetic.
Oh, absolutely. So we were hoping, good. That was our first hypothesis, right? So that would say that day 0 would be predictive of it, right? So I should have included that slide, but it shows that most of the prediction, 80% of the prediction comes from post day 8. So some things have happened post day. Again, it doesn't fully rule out what you're saying. Well, what manifests at day 8 could still be lingering at some day 0, right? So what we haven't done is done some attack-seek experiment, measured the epigenetic markers and things like that to completely rule that out. I don't disagree, right? So there could be some heterogeneity early on, which is kind of playing this role. So there are a few proteins which does it. So it's interesting. Another experiment that led to from was we were showing it to someone in Donnelly Center, and this person had this great intuition. They said, look, when you have, so apparently when you chop off your finger up to the nail bed, it comes back, right? If you chop off below it, it doesn't. So don't try it at home, but right. So the finding apparently is that the cells which allow this regeneration to happen have come from a lineage of cells which makes neural crest cells. So there's another organ which happens at early development, which allows for all the jaws and everything forming in the head. Those are called neural crest cells. So some of those neural crest cells actually give lineages to a lot of epithelial and mesodermal cells, right? So some of those cells seem to be important for this regeneration. So his intuition was, in these skin cells that you're pulling out, look at the cells which come from neural crest lineage, and they might actually be better at doing this. And that turns out to be correct. So you're onto something when you say, look, there is some hidden markings there, right? So they look like skin cells, but they all have different lineages that are merging into skin cells. So they remember where they came from, and some of that is super important to do that. But still, I think this highway idea is not a wrong one. Even if they knew where to start, they still have to know the whole path, right? Because this is a path in 100 dimensions, right? There has to be highways, otherwise it wouldn't work, right? Even if you got a good start, there has to be a track on which you roll. If there's no track, you kind of dead meat, right? So now we're looking for these trajectories, and well, making these trajectories is not we don't think it's super easy in this high dimension. So we've been developing some techniques. Well, now this is published actually. It's an older slide. So Spencer is a postdoc in the group who basically took this idea. So you can say, look, so how do people do these kind of things? I said this is all in gene expression space, right? So you can take cells at different time points, sequence single cells and their gene expression. This is very popular now and very easy to do now. And you can track them. You'll get data of thousands of cells going around in this process at different time points, and you will know what their gene expression is for 20,000 genes, right? I mean, 20,000 is a large number, and hopefully the dynamics is not in that. And particularly if you think they are pathways, you want to find out some low dimensional description of the process, right? So what Spencer did was basically took this kind of single cell data, which lives here, and design sort of a neural network basis like neural ODEs. I don't know if you guys are aware of those. So yeah, so she used neural ODEs to basically learn a dynamical system in a lower dimensional space, and use that to build trajectories that you can infer now, because you have a dynamical system, right? If you have learned a dynamical system, you can start it anywhere and it's going to run into a trajectory, right? So these are the kind of trajectories the system gives you. So here the cells are starting here. One trajectory is this, the other trajectory is this, right? So these are coming from her infer dynamical systems. And of course, they don't do very well in some areas when things are turning around and we're trying to figure out how to think about them. But so she spent a lot of time inferring these, at least developing tools to infer these trajectories. So this is sort of where our first, I'm going to end the stem cell business. Is this a good time to ask more questions if you guys have? Yeah, okay, good. So this was the story about, how non-genetic diversity plays out, one example of that. And the tumor talk I gave was also in the vein of this non-genetic diversity. So many of the ideas were getting actually developed in tandem when I was working on these two projects. So this helped us to think about how to think about what we were seeing in the tumors. As always, you never come up with a new idea. You just borrow an old idea and see if it works. So it's a classic case of that. But there it is even more surprising because it's tumors. So that kind of made it a little bit hard to swallow in some ways. But that's how we could explain the data. I'm not sure which. Okay, I'll talk about maybe microbes. If you have a choice between two, I can start with that. You guys care? No, okay. So I'll talk about phages and bacteria for a little bit. I just wanted to introduce viruses and phages in particular, right? So, well, phages are viruses which kill bacteria. Why should we really care, right? So there are many reasons we should care, but I wanted to give a very, very global reason. Again, if you look at viruses in terms of amount of carbon they have, it's tiny. Right? They are really minuscule in terms of how much footprint, if you think they have. Of course, we now know clearly post-COVID that they can be devastating in more than one ways. But that's true for bacteria too. But what is interesting in bacterial world is a little bit more. So if you really think about the food web, it starts at the bottom of the ocean. There are these autotrophs, right? This is the basis of everything. These are photoplanktons, which are converting light into food, right? They're taking dissolved carbon, nitrogen, sulfur, phosphorus, and making biomass, right? This biomass is eaten by grazers. These grazers are eaten by fish, and that's the seafood, right? So and up goes the value chain. And the heterotrophs are actually eating stuff that autotrophs produce. So these are the bacteria which don't consume light, but they consume secondary metabolites, which these guys are going to make, right? These could be interesting sugars, interesting, more complicated molecules. But what is crazy is in this chain, as the stuff goes up, 50 to 60% of it is just recycled. So 50% of these autotrophs and heterotrophs are killed by viruses every day. So none of that food is actually going up the chain. It gets made and then broken down again. So this is famously called the viral shunt. It was actually discovered like 10 years ago. Kind of crazy. It was done by a Canadian scientist, maybe 15 by now. But it's a very recent phenomena that we discovered that 60% of the food that is made is actually just churned over in the bottom of the ocean and doesn't even make it to the food webs. So it feels like a futile cycle, in the world of, in the world, the way we think about food webs. It's not clearly actually, what would happen if there was no viral shunt. So I think, it's a question for people who think about models and theories to really think about, is there a simple way even to think about what might happen? But what I want to highlight is this leads to, I mean, these numbers are kind of crazy now, like 10 to the 30 bacteria, a factor of 10 cycles, whatever. So you have tons of cycles every day, right? So it's not surprising that evolution really is looking for, there's a constant selective pressure for new things to evolve, because bacteria want to evade phage, phage wants to kill bacteria. So that's where really has been, this powerhouse of creating new and new biology. So anything you know of, CRISPR, DNA polymerase that we use for PCR, anything that we use in molecular genetics, all comes from bacteria and viruses. Every single one of them. Because they are really the playhouse when new biology happens every day. It's crazy numbers, right? It's sort of, for once one can say Avogadro number of things are happening, right? In the context of biology, right? So there's a lot of, and it leads to really, really interesting stuff. And one of the things that we went after was something which, working with Madeline, she wanted to think about adaptive immunity in bacteria, which is where the CRISPR system was discovered, by the way, right? So the whole CRISPR-based gene editing is what really bacteria uses to take a snippet of phage and remember it, right? And so what the game is, basically a bacteria would have a little memory of old phages. Different bacteria will have different memories because they would have seen different parts of the whatever they see. And what Madeline wanted to think about the dynamics of this problem. So here we didn't start with data. We just wanted to first write down a simple model to think about this thing and then go back and see if we can compare it to some data. So this is the kind of simulations I want you to think about, what she wrote down. So these are viruses. Those are the ones that mutate, right? If a bacteria remembers a virus, the only way a virus can infect it again is now it mutates a way that the bacteria can't use its memory to recognize it, right? Much like the flu. We get a vaccine, we remember all the strains of this year, so hopefully nobody gets flu twice, right, in the season. But by the next season, the flu has mutated away enough that our immune system cannot recognize it. It's the exact same conceptual problem, right? Which I think is the other reason of studying something like this, because you kind of have another way of looking at adaptive immunity. So this is how it looks like in a simulation. So these are phages mutating away. Bacteria can only remember something when a phage becomes large enough, right? If there are lots of phages, they're going to infect. It's a two-body problem now, right? There's an interaction to happen. So bacteria always is a sparse representation of phages. If some phages are just new phage came up, it never really grew too much, it's not going to get represented in bacteria, right? So you can see now these two populations co-evolving, if you will. So phages are sort of really properly evolving with mutations. Bacteria is kind of tracking it in real time, if you will, right? So can we see, so now you can see that in some ways this process is structuring the bacterial population, right? How many of them are different colors? Now you've structured this population, which earlier didn't have any structure, right? So can we look at this structure in data? So these are lab experiments, not done by us. There's a group in Berkeley who was doing those experiments, and they were measuring. So now these individual memories, they're called spacers, by the way, because the first time when they were discovered, they did not know what they were. They called them spacers. They were spacers between things which we thought were interesting. But importantly, the spacers were the most interesting part. So they're still called spacers, right? So you can track the dynamics of these spacers in time, right? And the simple model Madeline was drawing, you can actually start seeing, well, simulation-wise, you can see it, but you can do more statistics. You can see the turnover of spacers, right? So a spacer comes in, that phage dies out because it can't infect. New spacer comes in because new viruses are around now. So you can have this spacer turnover with time, right? So every other time you see, you're going to see new spacers in the population. So this is sort of the turnover time, fraction of the spacers of new types that goes extinct with time, so you can look at that. And these were sort of the spacer abundance distributions that you see. And this was also, it's kind of surprising that, at least in this talk, the theme seemed to be appearing. This is again the same kind of clone size distributions that I was making, right? These are rank abundance curves, but it's the same idea, where you have the spacer with the highest rank and so on and so forth. What is interesting is as generation goes on, early on, you see a lot of change in the distribution. But after, in this experiment, after generation 25 or something, those distributions actually become stationary in time, right? And Madeline's model could kind of recover those ideas, because what is happening is now you have established a traveling wave, right? So the phages are leading, the bacteria is behind it, right? Of course, they're turning over, new spaces are coming in, but now they're in this quasi-steady state where they're tracking each other with some mean distance, with some stochasticity. So the diversity that you have is the same, right? The shape of diversity remains the same. The spaces, the individual spaces are completely different at different times, right? The spaces here and here are different. But what do you mean? Oh yeah, like in the reprogramming problem, yeah.
So it's like the evolution requires some kind of very solid establishment, things that are not changing too fast.
I mean,
Like this
in some,
slice
yeah.
seems to be more important.
You can say that, but in this case, it really emerges, right? You don't put anything by hand, but you get these sort of diversity which emerges from these, this kind of dynamics. It's a dynamical property, right? It's not a microscopic rule. It's a dynamical property that emerges once you allow this coevolution to happen, because this is the kind of stationary state these things can go up to.
It's kind of like, it's all like.
Yeah.
The traveling wave thing you mentioned, it's like, you just see like there's a delay.
There's a delay. Absolutely.
The phage bacteria, but what's interesting is that they approach this stationary.
Distribution, right, right.
And
This is interesting, right? Because in some ways, what you're seeing is these two waves are now moving at some constant speed. No one is accelerating. The distance between them is not changing. So you have these two waves going about. So if you forget the translation, they're basically not moving, right? So up to this translation, they're not moving. Why this particular shape? That's an interesting question, right? Okay, I just wanted to show you a video of this. So here, the triangles are the viruses. You're going to see them run out, and then the circles are trying to catch them. So this is the kind of traveling wave you can see. This is from the same data I was showing earlier, and you can see sort of these triangles running off, and these circles kind of catching up to them. So this is the kind of traveling wave idea, and you can actually go and plot something slightly different. Now you can do phylogeny, right? And depending on how cross-reactive your memory is, right? So some vaccines are good because they cross-reactive and capture a lot of strains around that. Some vaccines are very narrow, right? That's why you keep getting COVID boosters, because they were not very cross-reactive. And flu, we do great. You take a vaccine once a year, the whole season is pretty good. It doesn't, our vaccine is cross-reactive enough. It also depends on how fast the mutations happen in that. But so you can actually get flu-like dynamics in these simple models that we draw. So this is the kind of phylogenetic tree you would see for flu if you plotted the actual data. I don't know if you have seen any. Maybe I should have, in this slide, I should include that. If you actually take flu data from 50 years or something since we have been tracking it, you basically see this kind of linear dynamics, because what we have is very cross-reactive. So the next thing that you can see actually has to have a big jump. And there are not many ways of getting a big jump. There's only one way of getting a big jump, right? So there's some successful thing that happens, and that spawns the new one. And then one of those successful things, goes out. But COVID basically looks like this. There's lots of things around it which are successful, right? So you can kind of basically have this weird sparse tree. So I think this is also interesting in context of this problem, because we're working with phages and bacteria. We can explain a bunch of data. And now we are really going into, so this was lab experiments. Now we are going into natural environments. So we have a group who goes and samples in Yellowstone National Park, these sort of hot springs, and where you have these phages and bacteria with the CRISPR system operating it full on. And we're trying to really understand the nature of traveling wave just from the data and see what it looks like. And what is interesting is we can really ask what different strategies bacteria use at different times, at different temperatures and different temperature gradients. Seems to be different things happening. So it's been a fruitful way of thinking about that. And I think a good conceptual model for adaptive immunity in general, even for human adaptive immunity. I think it kind of gives you a little bit more to think about. Okay, I have 10 more minutes. Okay. If I'm not drowning you guys completely with too much biology, I'll try the third one. In this one, we did experiments in my lab, so I'm always excited about it. So the idea here is, again, remember I told you each cell has many different mitochondria, right? So now you have the whole Darwinian dynamics happening inside a cell. But on the other hand, of course, if you have lots of mutant copies, the fitness of that cell also goes down, right? So there's a dynamics within cell. But then which one of these cells are going to survive, right? So when these cells are going to make babies, these babies, some are going to get only the black dots, some are going to get mixture of blacks and red dots. Now, what the fitness of these things really depends on whether they got good mitochondrial DNA or bad mitochondrial DNA, right? If mitochondrial DNA is bad in a cell, it's not going to function very well, right? So now you have a problem. If you look at this stage, it's basically a multi-level selection problem, right? So the selection has to operate inside a cell and across a population, right? And this multi-level selection has been one of the biggest sort of cornerstones in evolutionary biology to explain how multicellular organisms arose, right? Because now the selection is on the whole body rather than an individual cell, right? That's why we think how multicellularity evolved, or how a beehive evolved, right? Or an anthill evolved or something like that, right? Because the selection now is operating at the group level, right? But I find these yeasts provide a very good place to actually try and do experiments with something like this, right? Because you have multi-level selection, but you can do, it's very hard to, do experiments with anthills and beehives because you can't control any parameters. Here you can control a lot of them. So what Chris did was basically, is a student in the group who said, I'm going to understand at least this first part and then, deal with the second part. So his work was spent thinking about intracellular dynamics. So what he did, so this is how a yeast genome looks like. It's a 100 kb long thing. It has multiple genes. And if people who think about biology, they have their own translational code in the sense that, the triplet code that decides which amino acid encodes which triplet, mitochondria has its own different code than the nucleus. So they actually have to encode all the tRNAs and all that. So what is interesting is if you take a yeast cell, so why is a yeast a good system? So this is the baker's yeast, which we have all in our shelves, right, to make bread or wine or whatever. What they have is these mitochondria is 100 kb. If yeast cells can survive actually in glucose, where they don't need to use mitochondria for respiration, right? So they actually ferment. It's exactly what we want them to do. When they don't respire, they don't need mitochondrial DNA. So what they do is they degrade this mitochondrial DNA rapidly. And the way they degrade it is not by point mutations, they actually do recombinations. So there were old experiments which showed that if you take a yeast cell and grow up it in glucose, some of them will actually completely lose their all mitochondrial DNA. They don't even need it to survive. But as soon as you shift it somewhere else where they need it, they'll die, of course. But you can have these weird repeated structures where it'll take a little bit of DNA and just make many, many, many copies of it. Make a concatemer of this little piece. And the reason this little piece is so great at it, it has something called origin of replications. So now basically you have a piece of DNA which is just made of origins of replications. So this thing replicates way faster than the unmutated part and takes over inside the cell, right? So people knew about this in 70s, much of this field started in, but since 1990s, nobody sort of decided to look at the dynamics of the problem, right? How do you get from this thing to this little bit, right? What is the dynamics? What are the rules of that? And more importantly for us, what was exciting is, because between this and this, there are a lot of intermediate structures that can come up, right? Are all of them fitter? All of them have to be fit to be able to survive this. So we wanted to make a structure function type problem. So what are the dynamics? What are the rules of this competition? This is what Chris decided to do. And what he did was basically took yeast cells and just grew many, many, many of them in, simple glucose, right? So now all of them have some kind of error that they have made. but they all are pretty close to the original mother cell, which had the full genomes, right? So you can look at early dynamics of this problem, right? And then what he did was sequence all of them in, so this is how, so okay, let me just step back a bit. So now you have all these things, you extract mitochondrial DNA and sequence it, right? And because we wanted to find big structural changes, Chris started sequencing them in something called nanopore sequencing, where you can get 100 kb, 30 kb, 40 kb, really long reads, and look at them. So this is how a coverage of a good genome would look like. So it has, this is 80 kb genome almost. There's a coverage everywhere when you sequence. Some of the genomes would look like that. They've completely lost this part, right? And the amount between, for example, this part and this part is now can be a factor of 10, right? Because it has many more copies of this than this, right? So you can start seeing signatures of that, even if when you were not looking at structural changes. But so Chris sequenced, I don't know, 40 odd of them in different lineages, and you can see sort of all these guys came from the same lineage, so they're very similar. These one came from the same lineage, but this was earlier than this. So this came after this in some ways and lost more of this, and only this was left, as you can tell. So you can kind of start building now a story of how this might have happened.
And these are all like the same.
Same VAT. Yeah. So basically take something and just tweak them out, and now you're doing single, looking at single cells and single molecules in some ways. So let's look at something like this, right? So this is made up of many, many molecules, by the way, right? As I said, it's not one type of molecule. So now Chris was sequencing many different molecules, and what he was finding is you will find whole stretches, but you will also find sequences which only had this or only had this, something in the middle. So all these little bits were tiled in funny ways together, right? So you can find the frequency of all these different bits. You can infer all the frequency of different bits because he was sequencing these single molecules, just to give you a sense of how a single molecule looks like. So one of the molecules would look like that, for example. So you can see the repeated structure, right? The blues are repeated many times, the reds are repeated. So these are these 4 genes. So so it's this part. Where is it? COB. So COB is here somewhere. So it's COB, OE6, and a couple of other genes. So it's basically taking this bit and just making a concatemer out of it, right? So how does it make it? Remains an interesting question. So what we wanted to ask is, what are the features of this structure? So if somebody gave me a genome, can I say how replicative that is going to be, right? Because I get now quantitative numbers of different versions of those. So how do you find out how good a particular genome is? How do you measure its fitness? I know the structure because I can measure, I can sequence it. So this is again where yeast comes very handy. So yeast actually has sex, right? So you can take 2 yeast cells, the different types, and they can mate. So what Chris did was took cells which had this kind of DNA, mated them with cells which was wild type, which had the whole genome, and you can look at the progeny and find out how many of them got this. So it's a direct measure of finding out how fit this genome was. So he did sort of mating experiments. You can take these cells with red and blue, mate them, and then these mates will have babies. And depending on which genome is winning out, the babies will inherit more of one or the other, right? So he can now compute this fitness, if you will, of different petite families, right? So there were 9 families. In many families, you had different sort of lineages. Now he can compute the fitness of all of them. He knows the structure of each dot, and he knows the fitness of each dot. So some of the families have fitness differences which are pretty large, right? So this is that one family where you had, which lost a big chunk in its lineage, right? So it became much fitter than an ancestor. But the question now is, yeah, so for example, as I said, this one, the very fit one, is literally just has origin of replication replicated many, many times in its genome. That's all there is left, right? It's like a cancer. In some ways, this is like a genome cancer inside the cell because they can divide very fast. They lost all properties. They do nothing else for the cell, but they just divide very quickly. And you have all kinds of structures going on, at different levels. So, this one is this, and so on and so forth. So now the question was, can we make a model? Can we write down a structure function model between these two things? Because we have a function, which is the replicative fitness, and we have the structure. So can we figure out some relationship between that? So Chris came up with a model, and now he can take all that data and collapse it on one curve, right? Where he can assign sort of the suppressivity or the fitness of each genome and what its structure component is, and it kind of all falls on one line, where basically this in some ways is the density of these origins, if you will. It's a little bit more complicated than that, but it turns out it was very simple to understand, given some structure, how fit that thing is going to be. So where we want to go with it is, now we understand this part, right? We can play with lots of these parameters. We can play with how many copies you have, how fast the turnover. This selection coefficient is the suppressivity he was measuring, right? This is the fitness of each genome. So now we have all this sorted out, and we want to we can write down a simple equation to understand how many cells for particular composition would show up. So this is the intracellular dynamics. This is the extracellular dynamics, a cell level, if you will. This is within cell, this is cell level, and these are all the parameters we can play with now, right? So these parameters Chris has kind of figured out how to do. This one is something we are struggling right now, how to make a handle on. So, okay, it's a lot of biology, we can control these with some chemicals, we can control this by the whole library Chris has made of 40, 50 mutants now, so we know exactly what they are. This is what we can control with nutrient conditions. But it seems harder than what we were expecting. So we thought what we will do is give it a mixture of glycerol and glucose. Glucose is when it ferments, glycerol is when it has to respire. So we could just titrate these mixtures, and that will tell you how important it is for the cells to respire. But it turns out they don't really behave that way. They kind of switch around, and there's a time scale problem, and depends on whether nitrogen is getting starved or not. There's lots of other physiology which we are sorting out now to be able to control this parameter. So once we have this, there's a very simple equation. You can numerically integrate, you can do a bit of theory, you can do whatever you like. It will make predictions, right? You can have interesting qualitative predictions, like you might have a bimodal distribution, right? Because if stuff happens quickly inside, you'll either have blacks or reds, right? And then you'll have a bimodal distribution at one end. So there are lots of qualitative things this simple equation emits. We know simple equation like for fluid dynamics can give you very complicated stuff. So there's some interesting qualitative things we can check, and that's what we're hoping to do. I don't think I have time anymore. I'm running out of time. This is something we are starting to do now, starting to look at whole organisms, which is planaria. You can chop a planaria any which way you like, and it regenerates. So there's a couple of groups at EMBL we're starting to work with to try and understand that. Okay, I think I should stop here, and I mostly thanked most of the people I wanted to. Oh, this is some video of planaria. Okay, this is my group at some point in time, and Chris was here, Madeline, Sophie, and those are the people's work I talked about, and there was a bunch of others. This was the colleague who we sadly lost recently. So, dear picture to me. Thank you. Sorry, I downloaded a lot of stuff on you guys, but I hope at least the theme was a little bit clear of competition and how that plays out on these possible scales, different scales.
That was wonderful. Thank you very
Thank you.
much. I think I now kind of understand. I never understood why they study yeast so much for cancer, and now
It's amazing.
I have a much better
Oh, yeah.
understanding of how it's related. I know we did a lot of questions
Yeah.
during the talk. Does anybody have anything they'd like to ask now? Yeah.
No shortage of questions. So evolution can be understood as envelope condition or envelope pattern that I have to obey to between steps of any development. But what's the role of the adaptive to local conditions that somehow, in the end, converge to obey evolution?
Okay, so in first ways, I guess the first part of question is evolution is not very predictive, right? We did not know which next strain of COVID is going to show up, right? That's the part of evolution which is hard, right?
For the short.
Well, short, long, there's no idea which strains are going to survive, right? First, they're going to come up, what their fitnesses are going to be, right? So predicting evolution has been pretty hard. But what is interesting is, for example, for flu, we still have to make a vaccine for the next year, right? And making vaccine takes like 4 months. So we are kind of predicting what we think the next strain is going to look like, right? So while we can't predict, we still have to have some guess for what might happen, right? And that guess really comes from, because the way flu behaves. Flu has this very, linear jumps. So if we understand what the previous jumps were, something about them, it'll give us a bias at least of where the possible new jumps might be, right? So that's how we think about predicting. For COVID, it was super hard because it was just not doing that, right? It was basically any small change and its fitness could be very high or very low. So we had no way of predicting that, right? So that's one part of the answer of why evolution is hard, right? In terms of adaptive immunity, right? So what do you need for predicting anything? You need some idea of memory, right? There's something which persists in time, right? In flu, we know something persists in time, so the memory allows you to make a prediction possible.
Learning from history.
Yeah, something, right? So if the system has memory, depends on how long or small, you have a chance of doing some prediction. So adaptive immunity sort of does that for you, right? It kind of remembers the past infection. And if it is good enough to catch stuff around it, it's very good. It has predicted what things cannot infect you, right? So that's how I think about why adaptive immunity is more like a learning problem, a memory problem, rather than an evolution problem. If it was a purely evolution problem, it's completely chance driven. I have no hope of predicting anything. So, but this memory adds a different layer to it, which kind of turns the problem from, completely unpredictive to a lot of predictive power. Is that, yeah, maybe. So the memory part is the crucial part in this
Also
learning.
have memory less.
Not for predictions, right? For Markov model, you can't really, unless you know the probability distribution of outcomes, there's nothing you can do
So
beyond that, right?
in the literature, I don't have quotes, but some people, when they don't have anything else to say, they come with evolutionarily it must be so. So they invoke that
It comes from function.
as a closure.
Yeah, it comes from function, right? You say, look, birds have wings, they must be useful, and they happen to be, right? But many things are not. They're just spandrels, right? I mean, our appendix is a classic example of it. Lots of things are just spandrels of evolution because they're just there, because that's the lineage which kind of in which things happened, right? So, I mean, for example, tumor is a great example. You can take a tumor. So for a lot, I mean, it's still a problem, I think. You can take tumor cells, sequence them. You're going to find lots of mutations. You don't know which mutation caused tumor and which mutation just rode on that tumor, right? So these are called passenger mutations and driver mutations. So whole industry to find out what's a passenger mutation and what's a driver mutation. And I think we have like 3, like we have BRCA1, BRCA2, and maybe, I don't know, DMT1, 3 or something like that. We have like 3 examples after like 50 years of sequencing tumors, or maybe I'm exaggerating, but something around that, right? So I think that's what I would say to that.
Any other questions?
But tell me a little bit, so what excites you to do this? I know these guys, but what are your backgrounds, where you come from? Yeah. Okay, okay. Just want to, okay. Very good, very good. It's good. Fantastic. Matt, okay. Nice to meet you as well. Okay, good.
And yeah, it's one project I have is that genetic oscillations. in
Okay.
development.
Okay, great.
It seems that if the epigenetic factors oscillate, it stabilizes the development.
Okay. So it says early oscillations when sort of during early development, or what's
Yeah,
the,
yeah, it's really
cool? Okay, that's pretty interesting. Okay. Great.
Also noticed you had thank you very much. It was a great presentation.
Okay. Okay, great. Thank you, guys. Hope you guys something out of it.
Let's thank Sid.
All right. Thank you, guys.
Very much for coming.
Yeah, it's good. It's close.
Far.
Not at all.
Oh,
Very
okay.
fascinating.
It's on-
Different than at least what we
Yeah.
had before, so.
Yeah.
So yeah, some of the phage bacteria stuff another student is picking up, and he's doing more data analysis and things like that of these hot springs. So now we're trying to really make connection with the data on that problem.
I was thinking when you mentioned that it's also like in the soil.
Yeah,
that you have
everywhere, not
bacteria in the fat, you know, that's a hot topic now.
It's an interesting topic, but I think for me, it's important to find out a system where it's easy to study. Otherwise, it's just too much.
Yeah.
So I think this hot springs looks exciting. It's not that nice mesoscale of, not too complicated, not too
Right.
simple like a lab. So I think
And it's kind of like a little bit of isolated.
it's a little bit isolated. It's not constantly bombarded with everything. There's not so many hidden variables that we have no clue about and things
Yeah.
like this. Yeah.
On the picture you showed of the biomass, that
Yeah.
was interesting, I've never seen that. before.
Yes.
What are protists?
Protists are paramecium,
Oh, okay.
amoeba.
They're like their own little category.
Yeah, the grazers, right? So the one that
Okay.
they eat bacteria.
Okay.
Most of them are living right above bacteria. So if you go in any pond, it's filled
Yeah,
with,
yeah.
filled with protists, basically.
Very-
The eukaryotes. but
It's
not-
interesting about this viral shunt. To think
It's
about what would happen if
crazy.
that didn't exist, like we
Crazy. I've-
taken over.
Absolutely, it's amazing, it's, when I first heard about it, I'm like, what is this, And I don't think there's any even simple theory to understand what would happen if it was not there. I don't think anyone has even tried. Yeah, what's the point of it? And now that it is there, how does it inform everything? It seems to be important for almost everything that we see.
Yeah.
But on the other hand, it's some futile cycle if you want to think of biomass. It's sort of just
It's great.
cycling at the bottom of the ocean, making carbon to bacteria, bacteria to carbon. That's what it's doing. There's a lot of people think about it now much more like, oh, it dies earlier, sinks in. There's a lot of thing going on
Like
there.
helps the mixing maybe. in the
Why
ocean.
this could
Bottom of
be,
the
could be
ocean.
this, maybe, you can find some pattern there, I suppose. I don't know exactly how they think about it.
Yeah.
There's lots of work now.
So I did like long time ago, like fluid dynamics in the
Yeah.
ocean and how like
How it turns
this
out.
turning over
So you
because
can
of
play it,
solitary
yeah.
waves and stuff.
Interesting.
But maybe this viral.
Shunt
shunt
kind of,
is-
because there's a matter.
Because right at the
kind of-
bottom, there's not
Right.
much
Right,
motion.
interesting. Yeah, so it could play a role.
I can't believe we haven't met before.
No.
There might have been, like, when my first year was here in 2017, there was some committee about establishing like a biophysics program. And I think we might have been on that committee,
Maybe,
and then we
maybe.
just never met again.
Mathematics, right?
Yeah,
Yeah, so
Yeah.
there is some biophysics program now.
Yeah, yeah, yeah.
Exactly, yeah.
yeah,
Which.
For undergrads.
Undergraduates,
Yeah,
yeah.
because you have the grad.
Grad is, yes, whatever, grad
Very
students
big.
come in and work with whoever, right? I mean, there's no biophysics department. Physics department.
Oh,
In that
but there's
sense.
medical
There's
biophysics.
medical biophysics,
sorry.
which is its own thing, which
My
has been there
mistake.
for a long time.
Yes.
Yeah.
It's been there.
Oh, I know, I knew.
was late.
Oh, hi. Nice to meet you.
Good to see you.
Good to see you.
It was not the same talk, so.
I realized, yeah.
Sorry.
English is it, yeah.
No, no.
It feels from switching.
Well, it's recorded. It was
Yeah,
worth
I
watching.
know.
Very interesting.
How are you doing? Good, good.
I noticed on your website. Yeah, everybody's finding these out. Do you do aging work as well? Aging mice, I saw it on your website.
I'm thinking about starting to.
Okay. And what, like,
So there's actually, I'm going to advertise this. There's a colloquium next Thursday on physics, which is on aging.
oh, okay.
It's a guy coming from Dalhousie.
Can you
He's a
send me-
physicist. Yes, I'll
Is
send
it
you.
on Zoom or in person?
It's in person, but I think it'll also be streamed.
Okay.
So if you want
You
to
can
catch
send me
it.
the.
I will,
I'm
definitely.
interested in-
So yeah, that colloquium is interesting.
There is a
find it very-
delay in the stream.
They might
Yeah.
be, oh, so you've experienced this, okay, I see, okay.
You asked about it. I was
But-
wondering about the neural quiz.
Yeah,
So is this like stamping initial condition?
something like that. That's the only way to think about it.
And when you talk about the convergence of stationary distribution,
Yeah,
it's like things are allowed some freedom, and then in the end, what there is
yeah.
destiny.
But destiny in the amount.
In the, yeah, in
But
the
not in
amount,
what the
not in
things
what
are.
they are,
Yeah.
right? So you still can't tell which one is going to show up. What you can tell is in the density of things, right? It's sort of thinking like a Gaussian, like a central limit theorem or something like that, right? You don't know what you're going to get. In the end, the things are going to be distributed that way. But which one is going to be, there's no way of telling. Something like that.
Basically, in the game of life, there is a lot of degrees of freedom. But the end is somehow.
And all of them die.
quotes,
But
predictable.
something is
Yeah,
predictable. Yes, something is, some aspects of it are predictable.
Then I guess the question is, knowing that,
How does that help?
what
you
does
kind of-
that, hey, how does that help us to?
Correct, correct,
yeah,
correct, exactly.
That's a hard
exactly,
question.
some context. Like in
Trying
fluid
to
does that.
understand the steps.
Yeah.
Trying to understand the time steps.
Yeah,
Easier deal with
but
the
you can't
next
predict
step.
like, which one will.
Yeah, the
like, which
time-
one
yeah,
would
it's
die
some
out,
higher
which one would
resolution
take
thing,
over.
Some granular thing, not very small time stamp, some granular thing. So it's a very interesting flu paper, which you guys can look at. We didn't work on it, but a guy named Michael Lissig in Germany. They made a model of how to predict flu. And that's what WHO uses.
That's what they used to make, but I remember last year it wasn't very good, right?
No,
Was it last year?
huge, yeah.
They like
50% is
missed
the max you would probably get ever,
Yeah,
but it's still not.
but yeah, other years, I didn't get. Shot this year.
No, in principle, I think. I also avoid the COVID one.
Oh, yeah?
I got four. Four is enough.
For COVID, okay,
Not
that's,
that any Chinese fellow.
Yeah, I think I did get 4 as well. And I kind of stopped.
Four was enough.
yeah, thankfully now it
remember-
doesn't feel like it's not that sort of.
First one in Sunnyvale
Yes.
was
Oh.
Yeah, sorry, yeah.
so dramatic.
Sorry,
I'll use this,
At the start, yeah.
Okay,
Like
at some point.
The end of the world is coming.
So I'll leave this here.
Everybody was so serious and scared.